Niemann-Pick disease type C, or NPC, is a rare genetic disorder in children that we are studying in the Chang lab at Gaia through the inhibition of an enzyme called ACAT1. Shown here is an endosome, an organelle that sorts cell components. Shown in green are MPC1 proteins, which are thought to play a role in targeting and transporting cholesterol from within the endosomes to membranes and other parts of the cell. Organelles like the ER and mitochondria require cholesterol to function properly. NPC involves mutations in these NPC proteins that make endosomes unable to export cholesterol. This leads to the accumulation of lipids within endosomes as well as lysosomes, or organelle recycling vesicles. This results in too much cholesterol in some parts of the cell and deficiency in other parts. This gives cells an appearance called foam cells, which are particularly evident in the lungs, liver, and spleen. The pathology is observed in mutant mice with non-functional NPC1 proteins. The disorder is predominantly neurological. Although neuronal loss occurs throughout the brain, one of its hallmark pathologies involves the loss of Purkinje cells in the cerebellum, which is a focus of study in our lab. NPC does not currently have a cure. It primarily affects young children and leads to liver disease, dementia, neurodegeneration, and death. In the Chang lab, we are trying to reduce NPC pathology by inhibiting the gene for ACAT1. ACAT1 is a membrane protein located in the ER organelle that catalyzes cholesterol into a non-functional storage form called cholesterol ester. If we stop ACAT1 from carrying out this role, there will be more cholesterol in its functional form that could then be diverted to other organelles to repair cholesterol balance in NPC. In fact, in the past, the Chang lab has shown that when the ACAT mutant mice were crossbred with NPC mutant mice to produce mice with both mutations, these mice lived longer and had reduced loss of Purkinje neurons compared to those with only NPC. However, this isn't a method that could be applied to human patients, so currently we are trying to use a therapeutically applicable method to reproduce a comparable level of ACAT1 inhibition. One possible method is adeno-associated virus, or AAV-mediated gene knockdown. This method involves injecting AAVs that contain microRNA. Once they enter the cell, the microRNA silences the ACAT1 gene in the nucleus. Another method that we are more focused on at present is using small molecule inhibitors, which block the site where ACAT1 interacts with its substrates. The molecules are encapsulated in nanoparticles that are then injected into mice. Testing and improving the efficacy of this method is our current focus.